Monoclonal antibodies are incredibly specific tools. They target precise disease mechanisms without causing wide-ranging side effects. During the 2020 COVID-19 pandemic, the global demand for these large molecule therapeutics spiked massively. But you can't just mix chemicals together to make these medicines. They have to be grown. We use living mammalian cells as tiny factories, culturing them inside massive industrial bioreactor tanks. In the traditional approach, known as batch or fed-batch processing, these cells act essentially as polymerization catalysts. They sit in a closed tank and string together raw amino acids into long, folded protein chains. Because the tank is a closed system, everything the cells produce stays inside. As the population grows, they generate waste. Eventually, the osmotic pressure from these accumulating salts and metabolic byproducts becomes so dense it crushes the cells, and growth stops. By confining the process to a single, static environment, we place a hard physical limit on the maximum medicine a single tank can produce. Engineers realized that to grow more cells, they had to clean the tank while it was running. They applied a simple concept. The solution to pollution is dilution. This led to a method called perfusion. By continuously pumping fresh nutrients in and filtering waste out through a membrane, the cell populations can reach extreme densities, packing tightly together almost like organic tissue. But this creates a new problem. To keep this system stable and prevent the filters from clogging, operators have to constantly siphon off a percentage of the total fluid in what's called a cell bleed. Because the medicine is mixed in with that fluid, they are quite literally pouring a portion of the final product down the drain. To stop this waste, researchers developed dynamic perfusion. Instead of forcing the tank to maintain a perfect, infinite equilibrium, they allow the cell density to rapidly peak and naturally decline. Looking at this 16-day dynamic perfusion timeline, we see how it works. In stage 1, membrane fluid is sent to waste. When cells reach peak density, stage 2 begins. Saline flushes the bioreactor, pushing concentrated medicine out to be harvested. This dynamic approach quadrupled the physical productivity of the bioreactors. But pushing mammalian cells to these extreme densities triggered a hidden survival response inside the cells themselves. When we zoom in past the steel of the tank and look directly at the cell's metabolism, we find a biological phenomenon called aerobic glycolysis. When mammalian cells are forced into a highly proliferative, rapid growth state, their behavior shifts. Even with plenty of oxygen available, they start gorging on the available glucose and convert it directly into lactic acid. As this lactic acid secretes out of the cell, the pH of the entire surrounding culture drops. The environment becomes increasingly acidic, and the cells simply stop dividing. The very metabolic mechanism that allows the cell to grow quickly is the exact same mechanism that poisons the medicine factory. To solve this, bioengineers designed a feedback control strategy known as high PDOG, which stands for high-end pH delivery of glucose. This flowchart breaks down the mechanism. Scientists realized that if the glucose levels in the tank drop to a specific starvation threshold, the cells will panic. To survive, they reverse their normal behavior and start consuming their own accumulated lactic acid. As the cells eat the acid, the pH level of the tank naturally rises above a set point. A computer detects this exact chemical shift and triggers a pump to deliver a tiny microdose of fresh glucose. The pH drops, the pump turns off, and the cycle repeats. Researchers are now going directly into the genome to refine this further. By knocking out specific genetic elements like the BCAT1 enzyme, they can permanently disable the cell's ability to produce other obscure, growth-halting byproducts. By balancing precise starvation with automated feeding, the cells are coerced into an unnatural, highly productive state where they generate zero net lactic acid waste. Even with perfect metabolic control, these factories hit a strict logistical wall. If you start from a tiny vial of cells, it takes 2 full weeks of continuous division just to generate enough biomass to fill a single 13,000-liter production tank. To eliminate that waiting period, engineers split the process into 2 separate steps using a method called N1 perfusion, relying on a system of linked bioreactors. Instead of one giant vessel, a smaller seed reactor, the N1, keeps cells dividing at maximum rates. These multiply and continuously flush into the main production tank. Supplied with mature cells, it doesn't waste time growing, extracting finished protein immediately. By isolating the growth phase from the production phase, it ceases to be a stop-and-start batch process and becomes a high-speed continuous assembly line. Moving from static batch tanks to continuous perfusion systems and manipulating the metabolism of the cell itself has allowed engineers to systematically bypass the physical limits of mammalian biology. Decades ago, biotechnology relied heavily on painstaking, low-yield manual processes. Today, the extraction of these complex molecules relies on automated, hyper-productive manufacturing architecture. These engineering triumphs translate directly into volume. By keeping the cells alive longer and forcing them to produce more, the sheer amount of medicine available scales proportionately to the math of the bioreactor. As diseases require highly specific treatments, and as global health emergencies demand sudden responses, we finally possess the continuous manufacturing architecture to brew enough medicine